Oh, yeah. So let's wait. Let's wait.
2 minutes according to the standard.
Mm. I'm not doing like the bus we take in the morning. He waits 5 seconds and then leaves.
Yeah, we have 2 time zones here. One is there and one is there.
We still have 1 minute. My French colleagues are missing, so I'm waiting for them. Yeah, I should do that, yes. So, where are they? They're hiding somewhere. They're resetting, yes, that's the right word. Okay, so let's start adiabatically. So the third talk of this afternoon is by Sumantra Sarkar from Bangalore, and you will tell us about tissue solidification
Thank
through
you
cell
very
diffusion.
Yeah, thank you very much. Thank you, Sanjib, for having me here. Even though I just live 2 kilometers down the street, this is my first time giving a talk here, so it's a privilege. So I'm a biophysicist. I like to work on many different things in biology and biophysics. And because there was a discussion on how you can have diffusing diffusivity, in particular pathogen search by some of these immune cells. So this is a recent work that we have worked on understanding how T cell receptor actually searches for pathogens. And unlike the the diffusing diffusivity concept that was talked about in the morning, like there the switching happens between different modes of transport by these red things, which are the T cell receptors. Somehow the video got, okay, it's not working, but let me, there it is. So what is interesting is that, so you see these sort of green fringe-like things. So these are actin waves, which are formed by the the active nature of these molecules, and they form from the core of these T cells, these primary T cells, and they sort of move outward. So this is a form of an anterograde motion that is we are reporting for the first time. But what is interesting about this anterograde motion is that if you look at these particular T cell receptors, it is actually getting pushed by these anterograde waves outward, and what they are doing essentially is they are kind of looking for things over there, coming back, looking for things over there. They are doing something like this. And what is, yeah?
Those of us whose Latin is not so good, what is anterograde?
Yeah, good question. So, yeah, so anterograde means that you are moving from cell center to the cell periphery, and retrograde means you are coming from cell periphery towards the cell center. So people know about the retrograde flow of actin that is present, and it's some kind of a flow that it's similar to a hydrodynamic flow. And what this receptor does is it basically stochastically binds to either the anterograde waves or the retrograde flow, or it binds to none. It just basically locally diffuses around, and the transport is governed by this random switching between different modes of transport. Okay. But this is not what I'm going to talk about. My question today is a very kind of an age-old question, like people have been thinking about it for almost 20 years now, is whether epithelial tissues, the kind of tissues that we have everywhere, like our skin, like the lining of the organs, do they undergo a glass transition? Now, I probably won't have to introduce what a glass transition is to this audience, but for the sake of completeness, let me do so a little bit quickly. So we have learned about liquid-solid phase transition in our high school, and we know that liquid undergoes, if we are at a constant pressure, then liquid basically undergoes a phase transition to a solid through a discontinuous phase transition. There is a latent heat of freezing, which basically takes the liquid to a solid phase, okay? So what we did not learn in the high school is that this process is dependent on how fast and slow you actually cool the system. So if you cool the system slow enough so that you can your cooling rate is slower than the nucleation rate of the crystal, then you will discontinuously jump into a crystalline state. But if the cooling rate is fast enough, then you will get into what is called a supercooled liquid phase, which will eventually go to a glassy state through a non-equilibrium transition, okay? And over the years, people have found some hallmarks of this glass transition. And one of them is diverging relaxation time. The relaxation time diverges by orders of magnitude, like 12 or 14 orders of magnitude, for a change of temperature of by a factor of 2 or 3. There is dynamic heterogeneity, which means that you have coexisting regions of fast-moving and slow-moving regions. And also, you have something called aging, which means that some property, that measurable dynamical property of the system, actually depends on when you measure it. So these are not independent of the origin of measurement, okay? And some features of these have been identified and observed in experimental system in epithelia as well. So for example, here is a very famous example of the observation of dynamical heterogeneity in 2D epithelia. And what is interesting is that if you measure the average velocity over as the cell density increases through cell division, the average velocity decreases, and the size of this dynamical heterogeneity increases and eventually saturates to a maximum value as you increase the cell density. Slightly later, what people found out was that if you look at the relaxation of, say, velocity relaxation or anything, so there is a feature of something called jamming. And what is interesting is that these relaxation depends on when you measure it. So for example, in this particular experiment, if you measure the tissue at the 3rd day, you get a different response than if you measure it at the day 8. And finally, this is very peculiar for these epithelial tissues, is that if you measure the velocity spatial correlation, you find a non-zero correlation length, which is not what you find in an equilibrium system, which tells you that the glass transition or whatever transition that we are seeing over here is something that is happening out of equilibrium. And that is kind of important. So people have tried to understand this transition, this jamming transition or solidification through various theoretical approaches. And one of the earliest theoretical approach was to use this model called the vertex model. And people, in particular this group over here, what they showed was that if you measure something called the mean shape parameter, which is the ratio of the mean perimeter divided by the square root of area, there is a rigidity transition happening at a critical value of this shape parameter, which is approximately at 3.81. So crucially, in these particular theories, there was no accountability for the cell division, which happens all the time. These were all done in the absence of cell division. So the moment you add cell division to this system, what you find is that as the cell density increases, the system immediately fluidizes, okay? And people have tried to model the epithelial tissues using various different models, and they have always reported that cell division always lead to fluidization. And this has led these people to claim that there is really no glass transition happening in real tissues, and this is just an artifact of the experimental system on which they were observed. We, of course, don't agree with this statement, because in this talk, what I'll try to convince you is that there is actually signature of glass transition, and if you make some reasonable addition to the canonical vertex model, you can actually reproduce glass transition in theory as well, okay? So this is what I'll try to convince you in this talk. So with that, so this work was done in collaboration with Medhavi Bishwakarma's group at IISc, whose group did all the experiments, and Phanindra and Somodip from my group did the theory, okay? So let's go through with the first question, which is whether you can have tissue solidification through cell division, okay? And our tool of choice is the vertex model, okay? And the canonical vertex model has this energy function or Hamiltonian, which has this bulk modulus, and this is the perimeter elasticity. And also it has two parameters called the preferred area A0 and the preferred perimeter P0, okay? So these are convenient terms, but they are also unfortunately quite misleading because cells don't really care about a preferred area or preferred perimeter. What they do care about is something called contractility and line tension, which you can define in terms of these parameters. So what matters as a whole are the combination of these parameters, but not these individual parameters themselves. And in fact, in the original vertex model paper, they created a phase diagram based on this line tension, normalized line tension and normalized contractility, which showed that depending on the value of these two parameters, you can be in a different part of the phase. Okay? So for example, if you have low contractility and low line tension, you have a fluid-like network, whereas if you have high line tension or high contractility, then you have a crystalline state. Okay? So the question now that comes is, how do we relate this to something that can be measured in experiments? Okay? So through repeated observation, what we have found out is that contractility is related to something called the presence of stress fibers in this system. Okay? So stress fibers are long, fibrous actin filaments that are present throughout the cell. And the line tension is related to what is called the cortical actin or the junctional actin that are present in the cell. Okay? So if you have higher line tension, it means you have higher junctional actin, which implies you have high A naught and low P naught. Whereas if you have high contractility, then it means you have high stress fibers, which means you have low A naught and high P naught. And then, of course, for the sake of completeness, we also have this part where you have low A naught and low P naught, but they are hard to realize in experiments. In the rest of the talk, this particular picture is going to be really important, so I either recommend that you memorize it or take a photo if you want. Okay, so we are now going to use this picture to sort of argue that under what condition you can get solidification and why prior attempts were kind of not ready to give this kind of solidification from the vertex model. So for example, you can say that one of the ways I can model cell division is the fact that because the number density is increasing, I can assume that the area is also decreasing correspondingly, and I can sort of accommodate that area decrease by changing the A naught, which is supposedly the preferred area, as 1 by n. So this particular way. And I'll keep the P naught constant. So if I do so, then I get a trajectory like the following, where at low density, I am at this solid phase, and at high density, I am in this fluid phase. And basically, what that tells you is that if you increase the density, it will always lead to fluidization, which is what we expect. So, and in terms of this biological picture, we will start with a high junctional actin state and go to something which has high number of stress fibers. But if you look at the experimental data, this is not the case. So this is a data from 2015, and this is our own experimental data. And what you find here is that if you look at the system at low density, it has high amount of stress fibers. So for example, these kind of fibers you can probably see over here. And if you look at the system at high density, it has more junctional actin. So what that means is that the actual trajectory in experiment is exactly opposite of what we had like sort of predicted from looking at such a simple model. So what that tells us is that we need to modify the model, and one way to accommodate this kind of change is to consider something which is called the load-dependent binding of myosin to actin. Okay? So what this tells you is that the actin unbinding is actually not independent of the strain that you put into the system. The unbinding rate actually decreases with compression. Okay? So what that tells you, the more you compress the cell, for example, through cell division, myosin will bind stronger and stronger. Okay? And it has a nonlinear effect on the cell elasticity, which is what we should accommodate into our system. Okay? And to understand how this affects the cell mechanics, we first computed how the binding rate changes of the bound fraction of myosin changes as you change the cell area. And it gives you this kind of a hill-like form where A hat is a parameter which inversely depends on the myosin binding affinity. So the stronger the myosin binding, the higher the A hat. Okay? So, and this is how the P bound looks like. And what we know from the vertex model is that the line tension lambda is equal to minus gamma times P naught, and it should be proportional to the bound fraction of myosin. So what that tells us immediately that P naught should be proportional to 1 minus P bound, so P naught should increase as the area is increasing. Similarly, if you, with a slightly more complicated argument, you can actually show that A naught should be proportional to P bound. And if you take these two together, we have a constitutive relation which tells you that P naught should increase with increasing area and A naught should decrease with increasing area, okay? And what that means is that we now essentially have a mechanochemical feedback loop in which actomyosin binding leads to a positive feedback loop involving P naught and the cell area, and a negative feedback loop involving A naught and cell area. So what does that do is the following. The positive feedback loop leads to bistability in the cell area. So what that means, you can have both large size and small size which are stable. And what the negative feedback loop does is it doesn't let the small area go to 0 and the high area go to infinity. So it leads to some kind of stabilization into the system. And if you measure the contractility and tension, as you can see, the contractility decreases with increasing cell density, and the line tension increases with increasing cell density. And this is the kind of trajectory now we get from this particular type of constitutive law. And this is exactly what we found in the experiment. So then the question is, if we now use such a constitutive law and then run the vertex model, whether we can get tissue solidification. And this is the result. So here, as you can see, instead of fluidizing like before, it has become quite a solid, and there are, as you can see, there are large as well as small cells into the system, okay? So line tension is surface tension, because this is 2D, then you have to worry about the... Sorry. Yeah, so what is happening is that there is the actin. So the cell has a finite number of actin, and it is getting reallocated into two different morphologies. So there are these stress fibers, which are decreasing as you are increasing the density, and the junctional actin, which are increasing as you are increasing the density. So that's why the line tension is increasing, the contractility is decreasing. So which is kind of being taken into account by the constitutive law that you have, okay? Okay. This is simulation.
But how do you map cell to this kind of structure?
What do you mean? Sorry, what do you...
Physically there are cells, no?
Sure.
But cells are not lines. How do you map to this particular model? That was not clear.
So how do I map this particular model? I haven't really mapped yet. So what I'm going to do is basically, first I want to demonstrate that there is tissue solidification in the vertex model, okay? And then we are going to use that to compare with the experiment, because in experiment there is solidification. If we can't find that in the model, we cannot really argue that our model is correct? So first I want to demonstrate experimentally that we are getting similar type of behavior. So this is an experimental picture where, what I want you to notice from this video is that initially the tissue forms a soft network, just like our video, and then it eventually becomes kind of hexagonal and very crystalline. And cells are more motile at the beginning, and motility actually falls rapidly as you increase the density. And then also, just like the simulation, you have clusters of high density and low density regimes. And finally, the cells show some collective motion that seem to die as the density increases. So this is what you should observe in the experimental figure, okay? So you see that all of these high density regimes start to emerge, which seems very crystalline and very hexagonal in nature. And also the velocity, it kind of freezes into this kind of a very jammed cluster. So the motility actually decreases as you increase the density. So what are we able to reproduce just from this load-dependent binding of myosin is these 3 points. We cannot yet reproduce this, but in a bit, I'll show you how we can reproduce this. Yes?
What is the color code?
Yeah, so the color code is the size of the cell. So the red means large, blue means small, okay? And also there are some arrows which tells you that, or lines which tells you how fast they are moving, okay? So I hope what I have convinced you is that there is cell tissue solidification through cell division, what I have shown you here. So what we have to now convince you is what is the nature of this tissue solidification, what kind of thing is going on. And that is the next part of my talk. And what I'm going to convince you is that there is glass transition in this kind of proliferating tissues, okay? So
you have the heterogeneities in the previous plot, it was hard to figure out, yeah.
So why don't we actually see this? Yeah. You see, there are the small, large, small, large, small.
Okay, sorry.
Yeah, no problem. Sorry. Here? Oh yeah, so these are the regions where we could not track the cells. So there are cells, there are cells. You could see that there are cells. It's just that we could not track them using the best algorithm available. Okay. Okay. So to show that this is indeed glass transition, we go back to this picture and first argue that there is actually a discontinuous transition in this system, discontinuous freezing transition, that this tissue solidification that I have shown you has to be discontinuous. Without that, I can't talk about glass transition. Okay. So to do that, I go back to this picture that I showed you in the beginning, but now I be a bit more quantitative. So in this paper, from this paper, we have the equations that govern the phase boundary. So for example, this particular phase boundary is governed by the equation that the normalized tension is negatively proportional to the normalized contractility. So what we can now do is we can use the relation between the tension and the contractility in A naught, P naught, and gamma by K, and use this relationship, this constitutive law, to write down this lambda bar and all of these or this line in terms of the densities. Okay, because
Sorry, that's the previous question. So these cells you are looking under the microscope.
Correct.
And what do you mean by you cannot track it?
Yeah, so you are so the way these experiments work is the following. You basically look under the microscope, you take images at certain resolution, and then you have to put them under some algorithm to detect the edges, because these edges are very hard to detect. There are some noise, etc. So that's why the tracking algorithm, if they cannot detect the edges, they won't detect the cells. So that is the problem.
But if you see it, then you can...
Maybe we can, yeah, I can discuss it in the detail. So whatever edge we could detect, we showed. So it's kind of involved, so I'll talk later, okay? So similarly, this particular boundary has the following equation. So I can put that in there, and I can now write down this phase diagram in terms of the cell density, N, and this particular quantity, gamma by K, okay? So which is the normalized perimeter elasticity. And what is interesting here is that here you are seeing like there are two sort of transition happening from a fluid-like state to a crystalline state, and from a crystalline state to a polydispersed phase. And whether these are discontinuous or not is a matter that I show you in a bit. But what is interesting here is that here the cell density is actually playing the role of temperature in passive glass transition. Okay, so the transition is happening through density.
Number is conserved or
what number is conserved?
total number of cells?
Total number of cells. If you have cell division, the number of cells is not conserved. But in a bit, like in the next slide, what I'm going to show is we can do a simulation where the total number of cells is constant. So we can do a quasi-static simulation at constant density. Okay. So exactly that's what we are going to do here. So we pick this particular number because I don't really know how the phase transition is happening over here. We don't yet know. We know that there is a phase transition. We still have to work out. It's not entropic interactions. Yeah. But these are non-equilibrium systems, so temperature is not properly defined. Okay.
Those 4 root pi were secret numbers?
4 root pi is actually geometrically calculated. It's a secret number. Yeah. So let's look at this particular one, and we are going to do a quasi-static simulation, which means at constant density. And we measure something called standard deviation of trace sigma, which is nothing but stress fluctuation. So these are constant volume simulations, so we cannot measure volume or enthalpy as a measure of glass transition. So what we measure is stress fluctuation. If the stress fluctuation is large, then you are in the fluid state. If the stress fluctuation is low, then you are in the solid state. And here you can clearly see that at around this density, you get a precipitous drop in the stress fluctuation, which is telling you that there is a discontinuous change in the rigidity of the system, which is telling you that there is a discontinuous transition that is happening as a function of density. So this calculation is done in the absence of any motility, but we know in real system there are motility. So what we can now do is we can add a little bit of motility and see what happens. Oh, before that, so this transition kind of correlates with where people have predicted the shape index driven, the prediction of the transition. So wherever the shape index goes below this sacred number, we see that there is the discontinuous transition happening. Okay. So what is now interesting is that now if we add motility, this discontinuity actually remains, but just that the stress fluctuation becomes larger at some intermediate values of motility. But at high enough motility, you get this discontinuous transition to a solid phase and then again to a fluid phase, which happens through something like Kosterlitz-Thouless 2D melting process, where you accumulate loads and loads of topological defects, which melts the hexagonal crystalline state.
Just a reentrant transition.
Sorry?
Reentrant transition.
This is like a reentrant transition, yes, indeed, yeah. So this is what we have right now. So I have shown you that there is a discontinuous rigidity transition in this system. Now we can use that to show like cell density is defined. I have a constant box, okay, constant volume box. I just increase the number of cells.
But cells have different size, right?
Cells can have different size. So we start with all of them having roughly the same size, A naught equal to whatever is dictated by the.
So starting with
Yeah. And this is constant density simulation, so the number of cells are not changing. So all we are doing is we are basically now minimizing the energy, okay? So this is what we see when we do constant division rate simulation. So at higher division rate, I don't really follow this discontinuous change, but initially I remain in this sort of a fluid-like state, but then I kind of follow this trajectory that depends on the division rate. The higher the division rate, the quicker I fall into this particular trajectory, which is similar to what we see in passive glass transition, where higher cooling rate leads to quicker transition to glassy state, okay? What is more interesting is now we can also calculate the mean square displacement, which shows that at low enough division rate, you actually get diffusive behavior like a normal fluid. But at high division rate, you actually immediately go into a subdiffusive regime, which stays in the subdiffusive regime throughout. So what this is telling you quite, yeah?
Jamming picture, if you very rapidly pour in walls, then it will get stuck.
It's a quenching, right? So glass transition happens through that. Glass transition is a kinetic dynamic phenomena. It's not an equilibrium phenomena, right?
Yeah.
Yeah. It's similar to that, exactly that. Yeah.
And tell me please, at high densities, when you have this reentrant transition into
Yeah.
the fluid phase,
Yeah.
fluidization is caused by the fact that they have still different sizes.
They actually, so it is caused by the fact, yeah, so what is happening, is that the crystalline phase is purely like a honeycomb lattice, hexagonal essentially, and then you start getting this 5-7 defect pairs, and they start proliferating.
Okay, and otherwise you cannot accommodate them.
Exactly,
except
exactly.
to disordered phase.
Exactly, yeah. So we can also measure something called the overlap function and as well as the intermediate scattering function, both of which shows a plateau at high division rate, which tells you that this is truly a glass transition and not something else, okay? So, yeah, so this is the,
Features, for example, in your MSG plot,
yeah.
first ballistic and then plateau and then.
So this, you should not get any ballistic because this is overdamped dynamics. Yeah. Okay. So glass transition in cells or any cellular rigidity transition happens through shape. So we wanted to see whether we get similar transition in the shape parameters also. So one thing that we measured is called the asphericity, which you can measure by looking at something called the gyration tensor. And what we found the following is if you do this constant density simulation, which is quasi-static simulation, they kind of follow this particular trajectory. But if you do the constant division rate simulation, just like in the stress fluctuation case, you start seeing a very deviation from the quasi-static trajectory, and they go into a very different trajectory that depends on the division rate. So what is interesting is we can actually calculate the asphericity from experimental data by fitting an ellipse and finding the semi-major and semi-minor axis, which basically show that the experimental data also have very similar behavior when you measure the asphericity. So what this kind of convinced us that, well, from at least the static point of view, there is some kind of a signature of glass transition. So what we now wanted to do is to sort of look at some more dynamical feature like dynamical heterogeneity, etc. So for that, we come back to this picture, and I told you that there was this collective motion that we could not reproduce just from that one mechanochemical feedback loop. So it turned out that we had to worry about another feedback loop, which complicated our system a lot, but this was the minimal system that we could sort of devise. I'll discuss the details of this after the talk, but for the moment, let's take this into account. So what we have here is, in addition to this feedback loop, another negative feedback loop driven by this protein called ERK. So ARC is a Hopf oscillator which leads to some oscillation into the epithelia. It is kind of well known, okay? So if we incorporate that into our system, what we find is something really interesting, that we also now start to see this kind of collective oscillations and collective fluctuation, and even more so interesting that it kind of differentiates into two groups. In one group there is very fast oscillation, in one group there is very slow oscillation, okay? And what is even more interesting, we are also starting to get some kind of a collective behavior and jamming-like behavior. So we wanted to sort of understand whether we can reproduce the experimental observations that has been predicted some 20 years ago. So one of the first thing that we wanted to see whether we get dynamical heterogeneity, and just by comparing this data from Angelini, you can see that there are similarities, visual similarities at least. Then the question is, can we make quantitative match? To do so, we looked at these ones, how the size of the dynamical heterogeneity changes as a function of density and how the motility changes as a function of density. And for both of them, we see very similar behavior. So the dynamical heterogeneity increases and eventually saturates as you increase the density, and the motility decreases linearly, just like in the original one, okay? So these are a pretty good match, okay? So what is even more interesting, that coming back to this picture, you see that there are these kind of collective oscillations that we are seeing in this system, okay? And it is arising from the fact that each of these arc, the second feedback loop, leads to some kind of individual oscillations, which then get coupled to the entire tissue through mechanochemical feedback through cell-cell interaction, that lead to some collective oscillations which have much longer period than individual cells oscillation, okay? And you can sort of do the experiments, and you find that there is very similar behavior. When you look at individual cells, they have oscillations of the order of 10 or 20 minutes. But when you look at the collective oscillation in the tissue, they happen over the scale of 4 to 10 hours, okay? And we can compare our simulation with the experimental data, and we find excellent match between theory and experiment. And what is even more interesting is that we find that there is a collective oscillation damping as you increase the density as given by the time over here, okay? So this is kind of interesting, and we were not really, and this served as a kind of sort of a prediction and sort of test for the validity of our theory. But to understand why this is happening, why the collective damping is happening, we have to sort of go back to a much simpler theory that we devised some time back, okay? And to do that, we will try to understand how mechanochemical feedback can give rise to collective oscillation death. And it arises from something called PT symmetry breaking that happens in non-Hermitian system. To do so, we consider a 1D version of this system, okay? So in 1D, the epithelial tissues are nothing but springs that have some preferred length, L0, and some actual instantaneous length, L, okay? So instead of A0, P0, we have L0, and instead of the area, we now have the length of these tissues, okay? And each of these springs have this kind of mechanochemical feedback in which the mechanics, which is governed by the instantaneous length and the instantaneous rest length of these cells, is coupled to the chemistry of the arc, which is a Hopf oscillator, which we model here as a brusillator. What is interesting is that our choice of brusillator does not matter. So there is actually a theorem coming from group theoretic analysis which tells you that if you have a 1D ring, it doesn't matter whatever Hopf oscillator you choose. Any Hopf oscillator will give you identical result as long as you have same type of coupling. So instead of brusillators, if we have used Fitzhugh-Nagumo model, you would get exactly the same results as we have shown. Okay, so this is a general result. So what is interesting now is we can take this model. It is easy to solve and analytically, both numerically. And what we can do is study the property of an individual cell as a function of this parameter, which is the strength of the mechanochemical feedback. So alpha is the coupling strength between chemistry and mechanics, and beta is the coupling strength between mechanics and chemistry, okay? So what you find for an individual cell is that below this threshold, which is roughly about 1, you get a single fixed point, which is an unstable fixed point, and it is oscillating fixed point. Above this threshold, you have a saddle node bifurcation that gives rise to 3 fixed points, one of which is a stable fixed point. What that tells you is that if you kind of go into that fixed point, then you don't have any oscillation, which Hopf oscillators usually have. And this is happening because of the mechanochemical feedback. And it becomes even more apparent if you look at what the length of the cells are at that fixed point. And as you can see over here, the length is less than 1, which means the springs are compressed. And this is what we say is compression-induced oscillation death. Okay? So here the oscillation death is happening because the compression on the cell is leading to some kind of bifurcation, which is arising because of some complicated nonlinear bifurcations. Okay? So the next question is whether this survives for a 1D system. To do so, what we did, we went back to this particular model, and then we did a linear stability analysis of the continuum model that describes this system. And we found the linear stability matrix looks like the following. It has a block that describes the chemistry of the brusellator, which is very well known, and we know what happens over there. And there is a block that describes the mechanics of these springs, for which the rest length is also a function of time. And there are these two feedback strengths, which then govern the strength of the mechanochemical feedback. So if I set alpha and beta both to 0, which means that if there is no mechanochemical feedback, the mechanics and the chemistry are decoupled. And what is interesting is that the mechanics has something called Jordan two-block form, which means that there is an exceptional point when these two eigenvalues are identical. The eigenvectors then collapse onto each other. And that has some really interesting behavior. What is even more interesting is that because of this mechanochemical coupling, the exceptional point can sort of interact with the whole system and can give rise to a region where the PT symmetry is broken. Okay? And what we find that this PT symmetry spontaneously broken PT symmetry leads to 3 dynamical phase transition that give rise to 4 different classes of phases into the system. So over here, when the mechanochemical feedback is really weak, then you have chemistry-dominated patterns that you see for a normal coupled brusellators. Whereas when the alpha and beta are really strong, then you have mechanics-dominated patterns, such as traveling waves. In between, you start getting unique patterns that are never observed in any Hermitian system. So, and one of these is this collective oscillation death, and another is this bistable limit cycle. And this collective oscillation death is particularly important, and it kind of looks like this. On the bottom here, I'm showing you the chymograph of the concentration of one of the molecules. And on the top, I'm just showing you how it actually varies in space. So what you can see over here, there is this region over which there is no oscillation, but in the surrounding regions, there are oscillation. So this is what we call collective compression-driven oscillation death. And when you look at the mechanics of this system, you find that there is collective compression of all the oscillators in that region. Okay? And this is kind of interesting in the context of the 2D epithelial tissues, because as you are undergoing cell division, it is increasing compression on the collective scale. And because compression can lead to oscillation death, this mechanism persists even in 2 dimension, we argue that this is how oscillation death is happening in the 2D epithelial tissues. So with that, I would sort of conclude and sort of give you this particular picture in which we are proposing that epithelial tissues is actually undergoing a glass transition. And this glass transition is driven by the cell density. Okay? So if you do a quasi-static simulation where you are changing the density really, really slowly, you will undergo a crystal liquid and crystal discontinuous transition. But if you increase the density fast enough, such as through cell division, then you will undergo a glass transition. Okay? So with that, I thank you for your attention, and I'll be happy to take more questions.
Thank you very much for this very impressive talk. Deepak?
It seems like your morphology dependence on cell density is very weak.
Sorry?
You have to change the cell density by a factor 2 to see something significant. That seems surprising.
So which morphology?
In the patterns you showed about the cells below. Yeah, go left, go left.
Go left, go left. Okay.
Yeah, there's like these.
Yeah, so here the density is changing by a factor of 1.2. So at high density, they will just completely freeze.
Okay.
Yeah. So I'll show you. Yeah, it is changing within a factor of 1 and 1.2. Yeah.
So no transport-related results.
What do you mean?
Say diffusivity or anything like that.
So we have measured the MSD.
So from there, I mean, some plot of, say, diffusion constant as a function of your
These
density.
are so as I have shown you that right here, yeah. So for example, at high density, they always remain subdiffusive. So a diffusion constant is ill-defined in that case.
And tell me, do you control the thickness of the pattern?
Do I control?
Do you control the thickness of the pattern? Because sometimes
No, it's
you
two-dimensional.
Strictly two-dimensional.
Strictly two-dimensional.
Okay,
so,
so you confine it.
yeah.
Okay.
So so these tissues. So what happens? It's a very interesting question because so the experiments, these are done on MDCK cells, which are on a glass substrate, and they remain two-dimensional up to a very high density. If you go to a very high density, so for example,
you start to
5,000 cells,
thicken.
you undergo buckling.
I see.
Yeah.
Any other question?
So
Yeah.
does it matter when you start measuring your MSD?
Yes, it does matter. So at least, for example, the QT matters. I mean, we haven't looked at whether the MSD matters or not, but we definitely have measured the overlap function as well as the intermediate scattering function. They both depend on when you start to measure.
Yes, so you have aging.
Huh? Yeah,
You have
we have aging, yeah.
And you have a saturation at a certain point, or a crossover to a subdiffusion, or central.
Exactly.
Can you estimate how far particle or cell displaces up to this time?
So the MSD is of the order of, let's say here, 8 or 9. So and the cell length is of the order of 1.
Of one,
So 8 or 9 cell length.
so 8 or 9
cell length.
cells.
Yeah.
Okay. So it's quite far from the boundaries.
Yeah.
Okay.
So
Why does MSD saturate?
So the MSD saturate because it gets trapped. It's a caging. So this was exactly the question right now. So like, what is the distance? So the MSD is about 8 or 9, correct? It's not really much, whereas the whole system size is of the order of 40. So cells aren't.
Delta T behavior is not significant. It is low time, small time behavior.
Yeah. So the cells are moving like very so.
If it's ultimately caged,
Yeah.
I would not differentiate whether it is delta T or delta T square or whatever. Because ultimately it is caged, no trap.
Yeah, sure. So I don't understand the question then.
No, then what is the significance that diffusive? Diffusive in the long term?
Yeah, it's diffusive at short times. Diffusive at short times, so which means that
It doesn't matter, no? Small
shouldn't matter. So what I'm saying, so this is the T origin is 0. So I'm pretty sure if I measure the MSD starting from at a later time, I'll see that it will immediately remain subdiffusive because it will be caged at that time.
So that means you're already in the glassy regime, not that you are going to a crossover.
No. So this is not origin dependent. Like this is origin dependent. MSD should be origin dependent. This has been started at T equal to 0, and remember that this is only for the lowest division rate where our model remains fluid throughout.
For standard supercooled liquid,
Yeah.
you can use the
For supercooled liquid, it will remain in the fluid region. That's why it's a liquid.
it's already in the glassy phase.
It's not in the, so for this one, it is not in the glassy phase. For the high division rate, it is in the glassy phase. For the low division rate, it's not in the glassy phase. So the division rate is important. The higher the division rate, the more glassy it becomes.
And is the system uniform? So are there some regions in space which remain fluidized?
Exactly.
And you have a population of particles which
Exactly. So
start there and they move more or less
Right.
diffusive, but there are some regions which solidify or glassify
exactly.
already, and there they don't move at all.
exactly. So this is the dynamical
So you have
heterogeneity.
population splitting there.
Exactly. So you get coexisting regions of fluid and solid regions
Okay.
into the system.
So could there be some kind of biological consequence? For example,
Yeah,
cancer cells will be
Exactly.
more
So-
glassy than normal ones or less? I don't know.
I'm very glad that you asked. So basically, we have a separate set of experiments in which we try to understand why breast cancer cells behave differently from lung cancer cells. So lung cancer cells spreads, breast cancer cells form like a cluster, okay? And what we find over there is this kind of a glassy transition that is happening for the breast cancer cells, and it has to do with the differential tension between the healthy and the cancer cells. And it leads to some, I don't have the picture unfortunately here. So you can show that in case of breast cancer, you get a very compact region which basically then becomes jammed and glassy, whereas for the lung cancer, it remains fluid.
And ultimately, could
So
this help to
ultimately
detecting
what happens,
cancers,
yeah.
or-
So what are the consequences? What that tells you is that, like breast cancer, they are highly localized. So if you do some, say, local operation or something like that, you can extrude the breast cancer out of the tissue. For lung cancer, it is not so easy. It will just spread. Yeah.
Any other question? Okay, so let's thank the speaker again.
Yeah.
And the last speaker of this morning, this afternoon, I'm still jet lagged.
Thank you very much.
Is Giuseppe Del Vecchio.
I'm sorry.